Testing, testing. Okay, all right. Good afternoon to everyone. Thank you for sticking by. My name is Carlos Moya Villalobos. I'm from the University of Costa Rica, and there's like, yeah, okay, whatever. And I want to share with you a little bit about the work I have been doing regarding the molecular identification of endophytic fungi from epiphytic orchids of Costa Rica. Uh, where here? Okay. So as you may know already, orchids are one of the most diverse groups, plant groups in the whole world. They have been able to adapt to a wide variety of ecosystems, and they especially thrive in the tropics. And because of that, places in the neotropics, such as Costa Rica, for example, have been categorized as the diversity hotspots. These orchids form symbiotic associations that may have helped them adapt to a lot of different conditions and different environments. This, uh, this one here is Oncidium closcanum, one of the orchids from Costa Rica that I have been working with. So as I mentioned before, these orchids form a wide variety of symbiotic associations. One of the most crucial ones is the endophytic associations. So endophytes are considered all the microorganisms that live within a plant without causing any harm or any negative impact. In contrast to, for example, pathogens, etc. So there are three main categories of mycorrhizal of endophytic microorganisms, and the first one is the mycorrhizal ones. Mycorrhizals have for the most time been considered like one of the most important of these symbiotic associations, and they help the orchid throughout their whole life cycle. So, for example, they are essential for orchid germination and their early development. That happens because orchids have, as you may know already, very small seeds that don't have an endosperm, and thus they lack all the resources and nutrients and water in order to germinate. So what they do is they associate with these mycorrhizal fungi in order to get all these resources and be able to germinate. These mycorrhizal fungi form these very characteristic structures that you can see here that are called pelotons. And pelotons are like little groups or clusters of hyphae that form within the cell walls of the orchid. And the orchid, what does is they help digest these pelotons in order to get their nutrients. Most of these mycorrhizal fungi are classified in the Basidiomycota. And there are main key groups, all ordered within the Rhizoctinia group, which is a paraphyletic group. And they are mainly found in the roots and seeds of orchids, in contrast to, well, we will see later, but the second category is the putatively mycorrhizal fungi. And these are fungi that we think form these mycorrhizal associations, but we are not sure. So they stay as putative. Let's see. Yeah. And finally, we have the non-mycorrhizal endophytes, which are pretty much all the endophytes that live within the plant but don't form these mycorrhizal associations. And these, in contrast to the mycorrhizal ones, are mainly in the Ascomycota clade, and they can be found through the whole plant. So you can find them in the flowers, in the stem, in contrast to mycorrhizal that are found mainly in the roots. And some of these can be actually beneficial to plants in very different ways. So, for example, some produce antibiotic compounds that help the plant, and also they can produce phytohormones, and they can be helpful to the plant's development in a wide variety of ways. And some just stay there as commensals. In general, this endophytic group has been less researched than the mycorrhizal ones. So we know a lot less about how these interactions affect the plants. And well, in the recent years, molecular techniques have allowed us has provided us with different tools to identify these and study these endophytical fungi. And for example, the IDS sequencing has become like one of the most used techniques to identify these endophytic fungi. And as you may probably already know, the IDS region is is located between the small subunit and the large subunit. As you can see in this diagram, you have the small subunit here, the large subunit here, and you have the IDS region here. This region has two main characteristics that make it ideal for this kind of study. So the first one is that since it is in the ribosome, there are copies of it throughout the whole genome, and thus make it especially easy to amplify. And also that it is a relatively conserved region with enough variation to give us great resolution between different fungal taxa. It has maybe some limited resolution in some groups, such as Fusarium, but in general, this is the preferred technique for this kind of studies. So this work had like three main objectives. The first one of which is to isolate and culture all the endophytic fungi from these epiphytic orchids of Costa Rica. Then we wanted to molecularly identify these cultures by IDS sequencing. And finally, we wanted to create a database and collection of endophytic fungi of tropical orchids that holds all this information of this interaction between the endophytes and the orchids. The way we did that is that, well, first of all, we sampled in two sites, both pre-montane and montane forest between 800 and 1600 meters. And we sampled, as I mentioned before, the roots and the protocorms of several orchid species. These are the main genera that we have been sampled, so Oncidium, Masdevallia, Maxillaria, Epidendrum. And then we proceeded to culture the endophytic fungi in two different agar mediums. When the cultures were big enough, like they have enough mass, we proceeded to perform the DNA extraction with this kit. And then we used three different primer pairs to amplify the IDS region. So we used the IDS1 and IDS4, then the IDS1 and IDS4OF, both of which are like very general primers for fungal amplification. And finally, we used the IDS1 and IDS4, which is specific for the Tulasnelaceae family. And finally, we perform barcoding using Sanger sequencing to sequence the amplified regions, and proceeded to compare them to sequences found in the GenBank and UNITE databases. And we discarded all the sequences that shared like less than 90% identity. And in general, so far we have more than 200 cultures, of which we have been able to identify 94, and we have so far identified successfully 61 of these cultures. All of these are in 14 different fungal families, and what you see here is a little plot of the number of cultures per order of fungi. So, for example, you have Hypocriales, which was the most common, and with 22 cultures identified, then Xylariales, then Neotropical, and Agaricales, and so on. So, all of these fungi that we have so far identified have been previously reported, and we have found fungi in all the three different categories that I mentioned earlier. So first, we will start with the mycorrhizal fungi. We have found mycorrhizal fungi in Oncidium stenotis, mainly from the Ceratobasidiaceae family. These, along with some other fungi like Tulasnelaceae, have been some of the most frequently reported. And as any mycorrhizal fungi, they form pelotons with pelotons which transport nutrients and water. And there have been very recent studies that show that in Oncidium, the plant that we found this in, help promote germination and growth. And we think that perhaps testing new combinations of media will help us find more mycorrhizal fungi, but we have not tested that yet. Then we have the putatively mycorrhizal fungi. As I mentioned before, these are the ones that we suspect may hold these mycorrhizal relationships, but there's perhaps not enough evidence. So the first one is the Hypocriales, which, as I mentioned before, is the most commonly found. And it has been shown that genuses such as Fusarium form peloton structures that resemble a lot the pelotons we will find in these mycorrhizal fungi, and that Fusarium and Trichoderma aid in the plant development and germination, mainly through the production of phytohormones that break the seed dormancy. What is interesting about this group is that, in contrast to the most of the mycorrhizal fungi that we know of, they are actually Ascomycota and not Basidiomycota. Then we have the Agaricales, which have been often described found often in mycoheterotrophic orchids, and also that we know very well that they form another kind of mycorrhiza, ectomycorrhiza, with orchids. And so we suspect they may play a role in these endomycorrhizal associations as well. And finally, we have the non-mycorrhizal endophytes, which are, unsurprisingly, most of the taxa we have found so far. In general, they have all been commonly reported as endophytes in the neotropics. And as I mentioned before, they may have a lot of interactions with the plant that we don't really know of. Like, for example, they may benefit the plants through competition with pathogenic fungi. And lastly, I wanted to mention a little bit about why a collection like this may be important and enriching to these kind of studies. And the first reason is that even though 70% of the orchids are epiphytic and most of them live in the tropics, most of these kind of studies have been done in more temperate regions and in terrestrial orchids, which means that there is a huge vacuum of knowledge on these interactions in the neotropics. And secondly, that these kind of studies will give us a better understanding of the ecological dynamics of epiphytic orchids and the role that that microorganisms play in forming these interactions. So, for example, their distribution, competition with other orchids, and development may be influenced by these kind of relations. And thirdly, this knowledge will help us create symbiotic cultures that could help us conserve endangered species and propagate species of commercial importance. And currently, I just wanted to mention that currently we are starting these kind of trials where we co-culture mycorrhizal fungi with orchids, but it is still like very early on to have results. This is Oncidium stenotis, the plant in which we found these Ceratobasidium fungi. And finally, as conclusions, and take-home messages, I wanted to give 4. So the first one is that with the use of these molecular identification methods, it was possible to find a very great diversity of endophytic fungi associated to all these epiphytic orchids. And second one is that, as I mentioned before, this diversity is very comparable to what other studies have found in the neotropics. And third one is that these organisms, as I mentioned before, have a very deep relationship with the orchids and can have a fundamental role in their development, a lot of which is still unknown, especially in the endophytic ones. And finally, that a collection like this opens up a great amount of opportunities. So, for example, in academic research, as long as conservation, and also for commercial purposes. So, lastly, I wanted to give special thanks to my team, which have been incredibly helpful through this process. And that's all. So, if you have any questions, I will. Sorry. Yeah.
That was fast.
Sorry. You have more time to go to the gala dinner, so nice. That's a plus.
So if I understand, you're also culturing
Yeah.
what you find. So how much divergence is there between what you can find in the soil versus what you can culture? So
Oh, there's a lot. Like actually, one of the main, let's say, like bottlenecks of these kind of studies is that a lot of these endophytic fungi can be very difficult to culture in agar mediums, etc. So a lot of the diversity that you will really find in these orchids is lost.
Eh,
Yeah, you have. No, no problem, no problem.
So I noticed glomerales was on your list. Do you think that they may be?
Let's see, glomerulales. Yeah.
Do they ever make
Wait, sorry.
mycorrhizal associations?
No,
with
in
orchids?
general, they are considered endophytic fungi only. There's not enough, there's not enough information to confirm their status as mycorrhizal. So we mainly classify them as endophytic.
Any other
In orchids.
questions?
Yeah, in orchids.
No? Well, thank you very much all for your participation.
Mhm.